Good day, everyone, and you're welcome back to the channel. It's good to have you back here. Today, we're going to be discussing binary fission. Now, binary fission is one of the most popular ways, or one of the most well-known ways of asexual reproduction, and this is the way through which a lot of bacteria and some other organisms are able to divide. And in this kind of division, you have a single mother cell divided into two daughter cells, and these two daughter cells are exact replica of the mother cells. Now, it's the primary method of reproduction in bacteria, and it's a simple and a rapid process. It occurs very quickly depending on the kind of bacteria it is, and you have progeny. You have a mother giving rise to two daughter cells giving rise to four daughter cells, and it's an interesting process. Now, this process begins, that's the process of binary fission begins. There are quite a number of stages that occurs for the process to happen. Now, the first stage is the stage of initiation. Now, as the bacteria is growing, the bacterial cell grows, it matures, and then when it reaches this stage of maturation, there's what we call a growth cycle, a cell cycle, and then sometimes we call it a growth cycle too. And when this cycle is reached and the bacteria has reached full maturation, then there is a need for the bacteria to replicate. Now, for this bacteria to replicate, for us to have binary fission occurring, there is first the replication of the DNA, the single circular chromosome. We've said this in many of our other videos that the bacterial cell has a single circular chromosome. So this single circular chromosome begins to unzip from a point that is called the origin. Now, it unzips this singular circular chromosome in both directions. That is, it goes in opposite directions, and then when this DNA on the chromosome is unzipped and unwinded, and you now have two single template DNAs, you now have replication of each of those two strands of template of DNA strands. You have replication of these strands into new DNA, and that's how you have the new cell being formed. So this initial opening or unwinding of the strand is what we call the initiation process. Now, after the initiation process, we have what we call the DNA segregation process or stage. And in this DNA segregation process, an important part of the process is the formation of what we call a cell septum. This is a sort of wall that divides the two daughter cells. And for this wall to be formed, there is a protein called an FTS protein. Now, this protein, along with some other proteins that are present in the bacteria, help to form a structure that is called a Z ring or a Z ring. Now, this structure that is called the Z ring is the one that matures to form what we call a septum. And this septum is a wall, a partition, a division between these two daughter cells. So what you have is a mother cell where the DNA unwraps in the DNA initiation process. Now, after the DNA strands have unwrapped, you have these two different strands are pulled in two different directions, and then you have newer strands formed on those template strands, and then you have the formation of the septum, which is through the Z ring and the FTS protein, and that occurs in binary fission. Now, when this occurs, you now have, when the septum or the partition is well formed, you now have a complete division or a complete dissociation of the two daughter cells, such that they begin to stand alone as simple and different cells by themselves. And when they start to stand alone, then the process of cell maturation continues. Other biosynthesis begins to take place within these two daughter cells, and they continue to mature. So this process that we've described is a simplified version that explains to us what occurs in binary fission, where you have the division of a single mother cell to form two daughter cells. Thank you for listening to this video. I hope you got something from it. Have a wonderful day.